All right, functional annotation, lesson 6, getting up there in the lessons for functional annotation. So this lesson is going to be, we're going to talk about the KEGG database. We're going to talk about annotating with the various Koalas, the analysis tools that are provided alongside with the KEGG database, the Ghost Koala, Blast Koala, CoFam Koala. Those are really great tools, so I'm happy to chat about them. So why don't we just begin? All right, so KEGG. So KEGG stands for the Kyoto Encyclopedia of Genes and Genomes. So it's a well-organized database of data on genes associated by chemical pathways. And it's sort of presented in this really nice contextualized format that I think it makes it really intuitive and really easy to read and understand, especially for somebody like me when I was coming into it, didn't really know a lot about the various genetic pathways that there are. So if you're, it's a great way to explore your genomes if you can get it annotated with this. So the actual database was published in 1999. When it was initially published, there are 3 main databases included in KEGG. So it's been over 20. So the 3 databases are the pathway database, the genes database, and the ligand database. So the pathway database basically has a bunch of entries for pathway diagrams, and all of this was pretty state of the art in 1999. As you can imagine, over 20 years, this has changed a lot. There's been a lot more sequence information added to the databases, and consequently, they've expanded their sort of their databases that they have available. Okay. Right. So the pathway is the first thing I want to talk about here. So one really cool part of KEGG, apart from the organization of the genetic pathways, is as you can see here, the way it's organized: metabolism, genetic information processing, environmental information processing, cellular processing. These are really broad categories, and they're further broken down. So if you click on metabolism, you'll see that that's further broken down into lipid metabolism, nucleotide metabolism, amino acid metabolism, etc. And those are even further broken down into so if you click on energy metabolism, for example, you see oxidative phosphorylation, photosynthesis, carbon fixation, right? So if you click on these, you'll start to get down into the actual brass tacks of the genes, right? So I think that's a really cool, intuitive way of looking at it. Another cool feature is the visualization, which you can see here on the left side of this the slide. And this allows you to explore certain pathways. The one shown here is for glycolysis and gluconeogenesis. So these pathway components are all shown with these numbers. These are enzyme commission numbers, which you can click on and generate all the relevant information on the various databases. So if you, for example, click on this first 5.4.2.2, you can see that you get this info on the actual gene that's represented by this enzyme commission number, and all the pathways and all the databases that gene is part of. That's really nice. You get this kind of summary output. It's another example right here of nitrogen metabolism. So this might be more relevant to somebody that's more interested in environmental microbiology. So where you can look at the nitrogen cycling. So NAP-A, for example, here if you click on that, you'll see that the entries for NAP-A. NAP-A is a multi-subunit protein. So if you click on NAP-A gene, it's going to give you information for both NAP-A and NAP-B, and you'll get all the associated databases, all the pathways. Really nice. You actually get the reference here for that gene's function. So that's really well documented. Get more information, the enzyme, the reaction that this enzyme does. Right, so it takes nitrate and then it reduces it to nitrite, reducing water. Yeah, so that's really nice. Another really cool part is the feature that allows you to explore which pathway components are encoded by certain microbes. So, and that's controlled by this drop-down reference pathway menu. So if you click on that, and then you choose E. coli from a long list of bacteria here, it'll highlight in green all of the pathway components that are encoded by E. coli. So you can see that E. coli clearly doesn't encode all of the nitrogen cycling genes, but it encodes a few of them, including being capable of nitrate reduction. Off the top of my head, I don't really remember what all of these do, but I'm just going to move on. Glowibacter also doesn't encode nitrate reduction, but it has an RB, and you can see basically all the other components that it has, which are different from E. coli's. So anyway, that's all a really nice way of just exploring your genomes if you're interested in those kinds of pathways. So the KEGG genes pathway, these are the databases that are used in these analysis tools: BLAST Koala, Ghost Koala, and COFAM Koala. So I'm going to talk a little bit about these the web servers that I think are really intuitive and really easy to use. I was going to do a live demo of these, but I think it's fairly self-explanatory. But I'm just going to walk you through it. So first off, you have these three tools that you can sort of go back and forth between. So BLAST Koala, Ghost Koala, and COFAM Koala. BLAST Koala and Ghost Koala all were published at the same time, released at the same time. So BLAST Koala, so they're both, they do essentially the same thing. These are alignment tools, and once you upload your data and you upload amino acid sequence data to this web server, those sequences are compared against the KEGG genes database, and you can control which KEGG genes database you want to search against. So if you have bacteria or archaea, you choose the prokaryotes, or you could choose genus prokaryotes and eukaryotes if you don't know if you have both in your sample, or if you don't know what you have in your sample, or you could go even more broad than that and you can include viruses in there. So BLAST Koala is mainly designed for single genomes if you want to annotate a genome. If you want to annotate a metagenome, if you have a lot of data, you could use Ghost Koala, and Ghost Koala is a lot faster than BLAST Koala. So if you want to annotate a metagenome assembly, you can provide the ORFs, open reading frames, so gene sequences in amino acid format, to this Ghost Koala tool, and you can see that the file size limit here is pretty huge, so 300 megabytes. It allows you to submit. If you have more than that, you can break the file up and submit it in multiple runs. And then you just enter your email address and you click submit. Below there's a button to hit submit, and you get an email confirmation. So you have to go into that email and you have to click confirm, and then once you do that, the job's going to begin. And here you see that they show you how many jobs are in queue at the moment. So if there's a 0 like it does here when I took the screenshot, then and you submit a job, then you're basically your metagenome's gonna get annotated immediately, so you don't have to wait in queue behind somebody else that's annotating their data sets. I think that's pretty much all I want to say about this page. So GhostX, so the difference between Ghost Koala and BLAST Koala is that Ghost Koala, instead of using BLAST, it uses GhostX, which is another sequence homology search tool. I like this figure that they show. So they were able to show that GhostX is about 50 times faster than BLASTX, and above a certain e-value threshold, it's not that much less accurate than BLAST. So you can see that there are some pretty significant differences once you get to once you get up into the higher e values, like e-value 10 to the -5, you get a lot more false positives with these tools. GhostX is still pretty comparable to BLAST and 10 to the -5, but if you lower your e-value, if you up your e-value up to 10 to the -3, you start getting more of a false positive rate for GhostX, right? So once you get the results back from your BLAST Koala or your Ghost Koala run, and I'll talk about COFAM Koala in a few slides, but I just want to point out that you also, once you get the results, you'll get an email with a link that you can click on, and that link will take you to a page that will have either this result, if you submitted a BLAST Koala job, if you submitted a ghost koala job, you'll see this, and this is pretty nice because you not only do you get a functional annotation summary, but you also get taxonomy data for your submitted genes, and then you can download these data sets. If you click download, you'll get something that looks like this. It's actually not exactly what, so if you click download, you're just going to get the first two columns here. But if you click on preview first 100 here, you'll actually get a more detailed summary of your output, and you can still download from that page, and you're going to get something that looks like this. Later on, Ben is going to talk about the KEGG Decoder script that he has available on his GitHub, which takes this output and converts it to something that's a little bit more readable, user-friendly, and does some pretty cool summaries of that. A script that I have on my GitHub that I've sort of used from time to time, and I decided to make it publicly available, is this KO convert script. So this will take these files, these sort of raw summaries, and it doesn't matter if you download something that looks like this or just the file with the two columns where you see your gene sequence header in the first column and then the KEGG orthology number in the second column. It could be that or it could be this. And it will also take this KO orthology file where it lists all of the annotation and pathway data associated with every KO number. And then it creates a summary output that looks like this, the CSV spreadsheet where you have your header, gene sequence, you have your KEGG orthology number, you have your gene right there, and all of the pathways that the gene is associated with. So for example, the NADH oxidative reductases would fall under the metabolism, energy metabolism, and oxidative phosphorylation pathway. Actually, I should note that I got this KEGG orthology file from Elena Graham's GitHub page, where she has the Ghost Koala parser script that she created for the Onvio metagenomic workflow that allows you to actually take the Ghost Koala output and integrate it into your into the Onvio pipeline. So you could sort of have that in there in the profile database within that workflow. So that's a really nice use for this Ghost Koala results as well. Yeah, so that link is available on GitHub as well. So if you're interested in doing the Onvio metagenomic workflow and you want to, if you have a Ghost Koala, if you like the Ghost Koala annotation, you can include that annotation in your metagenomes. So CoFam Koala is the most recent addition to the KEGG orthology analysis toolkit. And CoFam is about 83 times faster than BlastX. Based on this paper, it appears that it's about twice as fast as GhostX. So it's comparable in speed to GhostX, but rather than using alignment, it is based on hidden Markov model, so HMMs. So what they've done is they've sort of compressed the genes database in KEGG into specific HMMs, HMM families, and those are used to search against a protein data set that you provide to CoFam Koala. So as far as the user concerned, there's really not much difference between these tools. You just have to provide gene sequences in the amino acid format. The only difference here is that you also have to provide an e-value cutoff for the HMM search that CoFam Koala runs to annotate your sequences. So I should note that when you are running Ghost Koala or Blast Koala, you provide your email. You can only have one job submitted at a time. So if you have multiple metagenomes that you want annotated, you will have to, and you have one email, like my Gmail, for example. Once you click compute, you're going to get a confirmation email, and after you click confirm and the job begins, you can't use this email to start any more CoFam Koala jobs, but you can use it to start a Ghost Koala job or a Blast Koala. So you can have one email per koala per time. So if you want multiple genomes or metagenomes annotated at the same time, you'd have to provide them with multiple emails, or just wait till it's done and do one at a time. Okay, yeah. Also, you're able to download the entire COFAM database. So COFAM are the HMM profiles for their KEGG orthology genus database, along with the predefined score thresholds. That's really cool. So if you want to do some local searching, if you know how to run Hummer and you have that installed, you can download COFAM. You can also download their COFAM scan, which is the actual script that runs behind the scenes of COFAM Koala and does the annotation. And this is what the output looks like. So once you get the email back with the results, you'll get a table that looks like this. And you can download it, and you'll get a text file. Okay, so I think that's it for my portion of the presentation, and I'll leave it to Ben to talk about KEGG Decoder.
Thanks, Arkady. Yeah, KEGG Decoder is a tool that I designed and never had any intentions of sharing with anybody. And then it slowly became more and more useful to myself and then the folks who are around, and they want to be able to cite it. So then it now is part of a paper that you can cite if you use it. It would be great if you could, and not a big deal if you don't. And so here's the web address to getting to KEGG Decoder. And so the idea behind this is that I wanted to take some of the default elements that you would get in something like a KEGG module pathway. So here's glycolysis. This is the same one that Arkady showed before. And process it in a way where we could quickly get an idea of how complete a certain pathway was based off of what was relevant to, if the pathway would work or not. And then generate that into a figure that will allow a quick assessment of if something had a pathway or not. And so KEGG Decoder relies heavily on the KEGG module components of KEGG Decoder, or KEGG Decoder relies heavily on the KEGG modules, but there's not exactly a one-to-one ratio. So some of these gene identifiers here are for eukaryotes, and being somebody who works with bacteria and archaea pretty often, those genes didn't particularly make sense, right? So there's been some curation of these KEGG modules to fit it into KEGG Decoder. And on the flip side, there are lots of elements within KEGG where the whole module might not be as useful as subsections of the module. And then on the flip side, there are definitely some metabolic functions in the environment that people are interested in that don't have KEGG models yet. And one of the ones I commonly point to is something like Animox, which doesn't have a KEGG pathway, it doesn't have a KEGG module, but does have KEGG KO values that you can search for in an output from something like Blast Koala, Ghost Koala, or COFAM Koala. And so KEGG Decoder does a lot at once and is somewhat simplifies some pretty complex pathways. And it's broken down into some very kind of basic organization to allow, once you kind of get used to what the KEGG Decoder output looks like, what might be present or not present. So on this side, we have core metabolism like glycolysis, the types of oxidoreductases you might have, the types of ATPases you might have. This is oxygen cytochromes here, carbon fixation, and all the pathways that we have assessments for carbon fixation. Some carbon degradation, a lot of these are just single presence absence values. Nitrogen cycle, sulfur cycle, hydrogen cycle, so the oxidation or reduction of hydrogen. Vitamins and transporters, not all vitamins, just a subset of vitamins. So methanogenesis is a little weird because and you need multiple components for it to actually function and actually generate methane as a biological process. These last two boxes right here denote the actual steps of converting something that has a methanol or methane group into methane gas. So it's the reduction of the COM cofactor, things like that. And then the compounds that contribute the methane are in this earlier part of this segment. So, there are some elements in KEGG Decoder that were designed for really personally for myself, that have now gone out there into the world and can cause some problems with folks. And then things like fermentation are checked. There's a whole section on secretion systems. And then a recent addition is a check to see if an organism can synthesize all of its amino acids based off of what's present there. And so there's a number of different visualization options. There's the one that we just saw, which is the static option, which just generates a heat map that you can use as a figure or make changes to. There's an interactive option, which will generate an HTML version where you can zoom or hover over features and it gives you a readout of a particular cell and which genome it belongs to. And then there's a feature that is still somewhat under development, but is currently coded in as part of the KEGG Decoder visualization called Tanglegram, which uses a Python library that creates these types of figures where you can give two different trees into it as an input, tanglegrams in general, and then create connections between where those components are on the tree. And for KEGG Decoder, what you can do is you can provide it with your KEGG output, so your Koala output from Blast Koala, Ghost Koala, or CoFam Koala. It'll generate one tree, which will group organisms by function, and then you can provide a NUIC file of the phylogeny of those same organisms, and it'll let them match up with these lines between them, how those groups are related to each other. And so this is a pretty busy figure with a lot of genomes, but this would allow you to look for discontinuities between phylogeny and function. And so with that, I actually have a binder tutorial for KEGG Decoder, which will be the hands-on component for this lesson. And so you can look for that video and join us there at the end of this video. Thanks very much.